So what if I told you that thousands of plants you see in nurseries, banana farms, even rare orchids, they are not grown from seeds, but they are actually clones, perfect genetic copies, and all of them started from something as small as a single cell. This is the story of how humans learned to grow plants without seeds, without soil, and almost without limits. In 1902, a German scientist named Gottlieb Haberlandt had a wild idea. He believed that every single plant cell has the power to grow into a whole plant. This concept is called totipotency. But here is the thing, he failed completely. His cells didn't divide, nothing grew, and for years people thought the idea was wrong. But guess what? Haberlandt wasn't wrong. He was just early. A few decades later, science finally caught up. In the 1930s, scientists like Philip White, Gauthier, Nobecourt, they started successfully growing plant tissues outside the plant body. And this was the birth of plant tissue culture. Instead of growing whole plants in the soil, scientists were now growing tiny plant tissues in the glass containers. But there was still a problem. The tissues grew, but scientists still couldn't fully control them. It wasn't until the 1950s with the discovery of cytokinins that tissue culture became truly programmable. And by 1958, Steward, using coconut milk, which has natural cytokinin, proved that they could regenerate an entire carrot plant from a single cell, providing the first experimental proof of cellular totipotency. And suddenly, scientists realized that if they changed the hormone balance, they could control what the tissue becomes. More oxygen, roots, more cytokinins, shoots, balanced, well, callus, which is a soft, undifferentiated mass of cells. But hormones alone were not enough. Plants also needed food, sugar, vitamins, minerals, basically a complete artificial environment. And then in the year 1962, two scientists, Morales and Skog, they developed what became the most famous tissue culture medium ever made, MS medium. And even today, most plant tissue culture labs they still use it. The medium contains nitrogen, phosphorus, potassium, vitamins, sucrose, agar. It's basically soil turned into jelly. And now, scientists finally had the right nutrients, the right hormones, and sterile conditions. Everything needed to grow plants artificially. This is where we get to micropropagation, which basically means growing many identical plants from a small piece of plant tissue. The process looks complicated, but actually very systematic. Step 1. Here the scientists first take a tiny piece of plant tissue, which is called an explant. It can be a shoot tip, a leaf, a node, an embryo, even pollen grains. And then the explant is sterilized because fungi or bacteria they can destroy the culture very quickly. So everything has to stay aseptic. That's why tissue culture labs look almost surgical. Next, the explant is placed onto sterile nutrient medium. Inside the bottle, cells slowly start dividing. Sometimes they form shoots directly, or sometimes they first become callus. Now comes the powerful part, multiplication. Here scientists they transfer the growing tissue into fresh media again and again for a limited period of time. Now each shoot here becomes multiple shoots, and those become more shoots, and very quickly one explant becomes hundreds of plantlets. This is why micropropagation is so efficient. A single edit plant can produce thousands of clones. Next, the shoots are transferred to media with more oxygen, usually hormones like IBA, NAA, and this stimulates root formation. Now the plantlets are complete. They have shoots, roots, leaves, everything. But still, there is one final problem. These plants, they grow inside perfect lab conditions. High humidity, low stress, no wind, no extreme sunlight. So if you place them directly outside, many die. That's why they go through a process called hardening, a slow adaptation process before entering real soil conditions. And only then they are ready for the field. Today, this micropropagation is everywhere, especially in crops that are difficult to propagate normally. Take bananas. Most commercial bananas, they don't even produce proper seeds. So how do farmers produce millions of plants? Well, you know the answer. Tissue culture. Same with orchids, sugarcane, potatoes, grapes, eucalyptus, teak. And because the plants are clones, farmers get uniform growth, uniform fruit, uniform flowering. It also helps to create disease-free plants, which is very important. And that's one reason tissue culture became a massive commercial industry. But micropropagation did not stop at cloning. It became the foundation of modern plant biotechnology, especially GMO development. And here is how. Scientists insert a new gene into plant cells, especially using Agrobacterium tumefaciens or a gene gun. But after inserting a gene, you only have a few modified cells, not a whole plant. So how do you turn one transformed cell into an entire genetically modified plant? Well, you regenerate it using tissue culture. The modified cells divide, they form callus, then shoot, then roots, and eventually a complete transgenic plant. Which means, without tissue culture, most GMO crops could not exist. Bt cotton, golden rice, herbicide-resistant crops, CRISPR-edited plants, all of them depend on regeneration through micropropagation. But this technology is not perfect. One issue is somaclonal variation. Sometimes cells change genetically during tissue culture, meaning the clone is no longer truly identical. This can create unwanted traits. Another challenge is contamination. A tiny fungal spore can destroy months of work. Besides, GMO-related tissue culture requires strict biosafety rules, containment, documentation. There are also questions about patents, ownership, genetic resources, because many tissue culture plants are commercially protected. So while micropropagation is powerful, it also needs careful regulation. And now, this micropropagation is entering a new phase. Automation, bioreactors, artificial intelligence, gene editing. Scientists are trying to produce plants faster, cheaper, and at massive scale. Some scientists are even combining tissue culture with synthetic biology, CRISPR, space agriculture. Because if I grow plants on Mars, they probably won't start with seeds. They will start with cells. More than 100 years ago, a scientist had an idea nobody believed, that one tiny plant cell could grow into an entire organism. Today, that idea feeds millions of people, protects endangered plants, powers biotechnology, and quietly shapes modern agriculture, all from a few cells inside a glass bottle. Micropropagation is one of those quiet revolutions. You don't see it, but it's everywhere, in your food, in conservation labs, in biotech companies. It's the idea that a single cell holds the blueprint of life, and if you give it the right conditions, it will build everything. So basically, we did not just learn how to grow plants, we learned how to restart life itself.